Let's see. Are we recording now? Okay.
Okay, yeah, I got a message.
Yeah, I got a message too. Okay, so everybody, we are being recorded, so everybody on their best behavior. And I'll go ahead, and if you see people coming in, go ahead and let them in. That's just for everybody. I'd like to introduce my friend, John Leo, who is a mechanical engineering professor at the University of Washington and works in their biomedical imaging I guess a group, and he's got a presentation on kind of an upgrade, as I get it, an upgrade to being able to do diagnostics and microscopy. So I'd like to hand it over to him, and thank you so much for being here, John.
Oh yeah, thanks for the invitation. Let me see. I'm getting a message. I'm trying to share my screen. So it looks like your audio driver. Okay, let me know if you have trouble hearing me. It's giving me a message that my audio driver is out of date, but I don't think that's going to be a problem.
Yeah, we can hear you so far. So,
okay,
well, you're not quite shared just yet.
Yeah, let me see if I can share just the presentation. I think you want to see my whole desktop. Well,
there we go.
You are seeing my whole desktop. Okay, let me make this full screen. Actually, let me try something else. This is a very large screen that I'm staring at. Can you still see that?
Yes, we can see that. Thank you.
Okay, all right. Well, I'll just stick with this. All right, so thank you for the invitation again. I'll be speaking about our work on non-destructive 3D pathology. I'll explain what that means, and we do quite a bit of analysis. So this talk is going to have two parts. One's going to be focused on the hardware, how we image the tissues, and then the second half will be more on the computational analysis, including AI, which is all the rage right now, as we all know. So I'm here at the University of Washington. I've been here since 2014. Prior to that, I was at Stony Brook on the East Coast for about 4 years. And then before that in the Bay Area for my training. I'm originally from Hawaii, so I'm definitely much more of a West Coaster than an East Coaster. So I have adjunct appointments in bioengineering as well as in the medical school in lab medicine and pathology, but primarily in mechanical engineering, and that's where all my degrees are in. So I'll give a broad overview of what we do in our lab first. We call ourselves the Molecular Biophotonics Lab, and we develop optical tools to help optimize patient decisions and patient care. For the longest period of time, we've been working on miniature microscopes for in vivo imaging. That means bringing the microscope to the living tissues, to the patients, instead of cutting out the tissues and putting it under a microscope, which is the standard way to do pathology. We miniaturize the microscope and either send it through an endoscope or a handheld device where we can image the tissues directly, non-invasively. And so this would be great for guiding surgical procedures, as well as for detecting diseases at the early stages. If we have a suspicious lesion, for example, in the oral cavity, that was one project we worked on, where it would be nice one day if every dentist had a handheld microscope where they can zoom in on these suspicious lesions and confirm if it's disease or not. There's a lot of lesions in our mouths that look like cancer. Most of them are not. Canker sores, cold sores could look like disease, but they're very benign. And so there is a need right now for tools to be able to help clinicians, physicians, as well as dentists to be able to screen for cancers. Today, I'm going to focus on the second topic, which is, we call it open top light sheet microscopy. That's the invention we had a few years back. And we use it primarily for what we call non-destructive 3D pathology, although we can also use it for guiding surgeries. So I'm not going to be focusing on that as much. I do have a conflict of interest to disclose. We started a company. This is a good thing. We're trying to get this out into the world, and we've sold a lot of our microscopes all over the globe, and trying to change the way medicine is done, trying to modernize the field of pathology. So this is led right now by the CEO is Nick Reeder. He was a resident here at UW in pathology when I first started in 2014. He was trained by a senior pathologist here named Larry True. They're both genital urinary pathologists, meaning that they focus primarily on the bladder and the prostate. And then Adam, another co-founder, was a former postdoc in my lab. And now he's at the Allen Institute, so he's still local. And so this company's out in Wallingford right now. So I'll start with a case study to illustrate the type of patient we're trying to help with our technology. As I mentioned, I've been working with a lot of these genital urinary pathologists who focus on prostate cancer. Men of a certain age, we are recommended to get our PSA levels checked. This is a blood test to look for an antigen that tends to rise when there's a presence of prostate cancer. If the PSA levels are elevated or if there's symptoms of prostate cancer, we usually go to see a urologist. There, the urologist in the US will take a dozen or more core needle biopsies. These are small 1 millimeter diameter plugs of tissue that are pulled out of the prostate, cut into thin tissue sections, stained, mounted on glass slides, and imaged by a pathologist using right now mostly a very standard microscope similar to the ones they use in high school biology classes. So this is technology that dates hundreds of years back, relatively simple analog light microscopes. One of the problems is there's a lot of disagreement when they're trying to grade the severity of the prostate cancer. Most prostate cancers are relatively indolent, meaning they won't lead to death. They won't lead to any side effects. But there is a small percentage, maybe a couple % of people with prostate cancer where that cancer could metastasize and could lead to lethality. And so the purpose of Gleason grading, the pathologist's job is if they see the prostate cancer, they have to try to determine, is this aggressive or is this benign cancer where we don't need to do anything? And so the Gleason's grading is based on the glandular structures. So there are these seminal vessels that run through the prostate. The shape of those branching tree networks of glands, it's like a vascular network, very convoluted. It's like a branching tree. The shapes of those glands, Don Gleason decades ago found that the morphology changes and is related to the aggressiveness of the disease. And so there's this scale that they use. Usually they don't use 1 and 2 anymore. It's really 3, 4, or 5 to describe these different patterns. So it's not a very granular scale. They're trying to approximate what they see into these broad categories of either Gleason 3, 4, or 5. In a common scenario, one pathologist may see entirely Gleason pattern 3 glands, which is a relatively indolent cancer. Another pathologist may look at the same slides and see a little bit of Gleason 4, mostly 3, but a little bit of 4. They call that a 3+4. Unfortunately, this slight difference leads to very dramatic differences in treatment recommendations. Gleason 3-3 usually leads to active surveillance. So you live your life with a high quality of life. You come back annually for additional biopsies just to track the disease. Of course, there is maybe a slight risk if this is truly aggressive cancer. It could lead to metastasis and maybe death. With Gleason 3+4, now they recommend typically surgical removal of the prostate or radiation therapy. It's about 50-50 right now between those two different choices. This reduces the risk of metastasis, but it could have potentially serious side effects like incontinence, impotence. So of the 250,000 men right now each year that are diagnosed with prostate cancer in the US, roughly half of them lie right in this Gleason 3 to 4 range. So it's a very common dilemma for patients and oncologists and urologists to decide what treatment is needed. So in summary, Gleason grading is not always reliable for distinguishing between lethal and indolent prostate cancer. And it's not just when the pathologists disagree. Even when they agree, the point is that Gleason grading is just an approximate guide. It's not going to be perfectly accurate for every patient. The one contributing factor is the way that pathology is done, as I mentioned, dates back over 100 years. You're fixing these tissues in harsh chemicals like formaldehyde. There's a lot I could talk about on that. it harms the DNA and RNA. It's not great for sequencing the nucleic acids, but it's also not a great way to preserve tissues dating back over 100 years. They dehydrate the tissues so that we can embed it into a wax block. This is simple candle wax, paraffin wax. That's in order that they can use a microtome to slice through the tissues. This is basically a fancy deli meat slicer, cutting the tissues into 4 micron or 5 micron thick sections that are mounted on glass slides, stained with most often this H&E stain, this familiar pink and purple staining pattern. This is a destructive process. We like to maintain those biopsies and valuable tissues for downstream assays, as I mentioned, sequencing and other assays. But this is a process that's slowly destroying the tissue as you cut through it. There's severe sampling limitations. They usually only look at a few thin tissue sections out of a biopsy or surgical specimen. That represents at most 1% of the tissue, usually much less than 1% of the biopsy or surgical specimen is actually seen. So you're missing a lot of the tissue and you're seeing it in 2D, and we of course live in a 3D world. So the vision is, if we can find better ways to preserve the tissue and to clear it, I'll talk about that later. Clearing makes the tissue transparent so that now we can image it in 3 dimensions with a light microscope. And I'll talk about our technology for doing that. The advantages of being able to do this is it's entirely non-destructive. We can image through 100% of the biopsy comprehensively without destroying any of the tissue. So we can give that tissue for other scientists wanting to do other types of tests. And we can image everything in 3 dimensions. So a question we often get from pathologists, we don't get this question from engineers.
All you got to do is throw it in a, go for a basket.
Hey, can we have everybody,
What's
sorry
McCall,
about that.
so-
Can we have everybody put on their
no
mute,
problem.
please? Sorry about that, John.
No problem.
Get that
Okay.
degassing problem or
Oh.
something like that.
I wonder if we can
Is
mute
that
others.
Scott, any chance you can mute yourself there, buddy?
Okay, I'll continue. So, like I was saying,
What's this now?
yeah. Oh, okay. Hopefully, we'll be able to hear me. The pathologists, because they're so used to looking at tissues in 2 dimensions, they often will ask, well, what would be the value of 3D? It's sort of obvious to most people that 3D would be better. But here are some examples, as I mentioned, with prostate glands, like similar to blood vessels, they exist in these very convoluted branching tree networks. The 2D views, as I'll explain later, I'll give some examples, can be very misleading. And so being able to image this in 3 dimensions should be a lot more informative and accurate. Another example would be looking and quantifying complex distributions of cells. One example is there are a lot of immune oncology, immunotherapy methods that are being developed now, where people have shown that if you can quantify the relationships between the tumor cells and the immune cells, you can predict which patients will respond best. And these are expensive drugs, sometimes as much as $1 million per treatment for these cancer patients. Insurance companies certainly don't want to be paying for drugs being used on the wrong patients where they have no effect. A lot of these immuno-oncology treatments only work for about 20% of the population. And so that's another big market where if we can image these cell populations in 3 dimensions and start to quantify the relationships, we can develop methods to predict which patients would respond the best. And finally, sparse or rare objects. There are objects that pathologists will see occasionally. For example, tumor cells that are starting to invade into the vessels or lymphatics. Obviously, that's not a good sign because it's going to start traveling through the rest of the body. There are things like stem cells and residual disease after surgery or after chemotherapy that are rare and hard to see on 2D sections because they're looking at so little of the tissue. If we can image a large 3D volume of tissue and apply AI tools to find these rare events, that could help with a lot of treatment decisions and patient management decisions. So in terms of imaging tissues in 3D at high resolution, there's a few ways we can do that. One is a sort of brute force serial sectioning method. If we can cut through the tissues slice by slice, this can actually be done fast. It's been commercialized in the past with these robotic sectioning devices, but it's destructive. So it's not something you want to do on valuable clinical specimens. Another approach would be to use what we call laser scanning microscopy, for example, confocal microscopy, two-photon microscopy. These techniques have been used in the life sciences for decades, and they work very well, but they're reconstructing a 3D volume essentially point by point, or at most line by line. And so they tend to be slow and complex. They have to scan this focused beam in a raster pattern or some other predictable pattern and reconstruct the image point by point. So not ideal for clinical specimens that are quite large. We need it to be high throughput. We need to be able to image many specimens quickly. And so about 15 to 20 years ago, people started exploring light sheet microscopy. And the concept is quite simple. Instead of cutting the tissue into thin sections with a knife, if you can make the tissue transparent, you can send in a sheet of light to illuminate the tissue from the side. And so this is literally just a sheet of light that's very thin, about a few microns thin. It illuminates a thin section of the tissue. It excites the fluorescence as long as you've stained the tissue with fluorescent dyes. Now you can image it orthogonally, just like you would image a glass slide, onto a very fast, sensitive scientific CMOS camera. So this is camera-based imaging. We're collecting these full camera frames rapidly, and as we move the light sheet through the tissue, we can rapidly image in 3 dimensions. So pretty simple concept. The early light sheet microscopes were designed, though, for these model developmental biology organisms like embryos and fruit flies and Drosophila, zebrafish. So they were not designed for clinical applications. The sample prep was very tedious. They would mount these tissues in agarose gels. And these microscope objectives were quite bulky, limiting the size of the specimen and the numbers of specimens that you can image. You have an illumination objective arranged orthogonally to a collection objective, and the specimen would be placed within a cuvette or a capillary tube right at the center. So that was very low throughput. So back around 7 or 8 years ago, we were thinking about this problem and had the concept of, couldn't we design something like a flatbed document scanner for tissues, where it's as simple to use as throwing a tissue on top of a glass plate, or it could be a modular well, multi-well plate, or a customized biopsy holder, but very simple, open top design where all of the optics are underneath, just like a flatbed document scanner, and we just place the tissues on top. So we did that. We sent in the light at 45 degrees, and we collect that orthogonally. There's another 45-degree light path to collect the fluorescence light out onto these high-speed cameras, very sensitive scientific CMOS cameras. I won't go into too many details. The challenge with this design is really getting the light in and out of the tissue without being aberrated severely. So if you have angled light focusing into tissue from air into a high index glass and into the high index tissue, it tends to aberrate. There's a lot of refraction. And so in our first generation system, we had this hemisphere that provided a normal interface for all the rays of light as they transition from air into this high index glass. And then we do index matching, meaning we use a thin layer of oil with the same index as the glass, and the sample is sitting on top of another glass plate, also with the same refractive index. That allows us to get the light in and out without being reflected or refracted or bent or aberrated in any way. This is a stage scan system, meaning the light sheet is stationary in space. In order to create a 3D volume, we move the sample with this stage. This is a 3-axis sample stage. As we move the sample, for example, from left to right, we collect a series of these 45-degree angled light sheet images from the camera. And we can reconstruct that into a volumetric brick of data. And we can scan adjacent bricks to image a larger volume over time. So again, the open top gives us that ease of use for a clinical setting, and the light sheet gives us the speed that we need for large clinical specimens. So our second generation design allowed us to lower the specimen. If you look at this first generation design, we had this solid piece of glass that prevented us from lowering the specimen if we wanted to image deeper within the tissue. So with the second generation system and all of our subsequent systems, we have this immersion bath. This could be an oil bath or a water bath where we have this one immersion objective. Now we can lower the specimen by about 3 millimeters in this case to image deeper for larger specimens. But otherwise, it operates very similarly, where we collect a series of these 45-degree angle light sheet images that gives us that volumetric brick, and then we can image adjacent bricks over time to create a large volume. And so this video is showing how we scan the specimen strip by strip, tile by tile. Each time we scan in one direction, we're collecting basically a brick of data, a 3D brick of data. One of the challenges that I mentioned is index matching. So as we increase the resolution of these devices, as we try to increase the focusing power, we call that the numerical aperture of these beams. Basically, it's the cone angle of this collection beam path that's shown in green on the bottom right here. As we increase that to be able to focus more sharply and see smaller structures, we're more and more sensitive to the index mismatch between the immersion bath, the sample holder, and the tissue itself. And so we start to get aberrations unless we can make sure that all of these materials have the exact same index of refraction. So that becomes, I'll talk about that a little bit later, that becomes a little bit of a challenge. Our third generation system was our most compact system. You can see we can package things to be the size of approximately a LaserJet printer, about 1 foot by 1 foot by 1 foot footprint. Here we have our first multi-resolution system. As you remember from using microscopes, even in high school biology classes, there's a turret of objectives so we can zoom in and out. That's important in three-dimensional pathology because the imaging times and the data set sizes scale to the third power with the resolution, the magnification that we want to image the tissue at. So we need to be quite judicious in the level of magnification. If we want to image large amounts of tissue at high resolution, that takes time. But we can first scan things at low resolution, and then once we find the regions that we want to zoom in on, we can switch the microscope objective and then image that small region at high resolution. So it's some sort of a multi-resolution workflow. This is our latest system. We call this our fourth generation hybrid system, where, as I mentioned, one of the issues was the index matching of the specimen with the specimen holder and the immersion bath. And so with this latest system, as you can see, the collection objective is no longer at 45 degrees with respect to the specimen holder. We're orienting the collection objective vertically. So that removes a lot of these off-axis aberrations that were plaguing us before. So this system's a lot less sensitive to the index mismatch requirements of the previous systems. It also has one of the higher NAs, a higher magnification than our previous systems. Also, by arranging this objective vertically, we have access to the full working distance of this objective, meaning we can image more deeply. If we angle this objective at 45 degrees, we will lose some of that distance because of the bevel of the objective, as well as the 45-degree angle coming in. And so this particular objective, our latest objective, has a 1 centimeter working distance. We can image, for example, whole mouse brains are placed on our specimen holder. We can image through an entire mouse organ or large clinical specimens. It's also a hybrid system, meaning that we still have our illumination path here in our collection, our main collection path vertically arranged, but we have room for a second collection path if we want to do low resolution imaging. So within one system, we have 2 beam paths, one for low resolution imaging along the sides, that's our traditional orthogonal arrangement. And then for high resolution imaging, we use this, what we call this non-orthogonal dual objective arrangement. And so you may be wondering, how do we image a light sheet in the orthogonal direction if we're not coming in at 90 degrees to that light sheet? And so there's a little bit of a trick here where we set up a relay. This is basically creating a one-to-one replica of the light sheet to this remote focus. So this is just a relay system where we can bring an image of this light sheet down to this remote focus point. And then we can tilt our camera arm so that we're imaging that light sheet in the correct orthogonal direction. So it's a bit of a trick that we borrowed from others to make the system work in this multi-resolution mode. So here's just a summary. There's a lot of numbers here, but the basic take-home message is that as we have developed different generations of our systems, we've improved the resolution. Now we have multi-resolution systems that can image at high resolution as well as low resolution. Of course, imaging speed, this is a log scale, so imaging speed trades off severely with the resolution. So we have to be careful with how much resolution, how much magnification we want to image at. Our latest system has the longest working distance. As I mentioned, we have a 1 centimeter working distance, so we can image quite thick specimens that are transparent. And then this shows us the maximum tolerable index mismatch. So with our first 3 systems, as we improved the resolution, we got more and more sensitive to the index mismatch that I mentioned earlier. With our latest system, because we have that non-orthogonal arrangement, we're a lot less sensitive. So this improves the ease of use of our system. And now we don't have to worry so much about index matching. I just have one slide on tissue clearing. Over the last 15 or so years, there's literally been hundreds of papers published. It seems like magic to make tissues transparent. It's actually not hard to do at all. One of the simplest ways to make tissue transparent is to remove the water and to replace the water with a high index oil. Water is the biggest culprit. It has a pretty low index of refraction of 1.33 compared to the proteins and lipids in our body, which have a higher index of about 1.5. And so that's the reason why a glass of milk is opaque, whereas a glass of oil is transparent. Because in a glass of oil, everything has the same index. It's homogeneous. So as the light goes through, it just passes right through. And there's no refraction, there's no reflection. With a glass of milk, you have a lot of lipid droplets surrounded by water. So every time the light travels and hits an interface between water and oil, some of the light bends, some of the light reflects, and it makes it an opaque liquid. And so that's exactly what's happening with tissues and why they're not transparent. So our favorite approach, simplest approach, is just to dehydrate the tissues and replace the water with this high index cinnamon oil, ethyl cinnamate, which is very safe food grade cinnamon oil. There are ways to do it better. Some tissues are harder to clear. They're maybe lipid-rich, fatty tissues. You can even clear bone. You can decalcify the bone. Some of these approaches require removal of the fat, delipidation, and then you can make the tissue very homogeneous in the index of refraction. So there's been a lot of work that's been done in that field. A lot of our tissues are stained with what we call a fluorescent analog of H&E. And that's intentional because H&E histology is what pathologists in the hospital look at 90% of the time. It's still the standard of care. It was developed over 100 years ago, and it's really the basis, the gold standard for how they diagnose tissues in most cases. And so our microscopes are fluorescent microscopes where we can try to replicate the appearance of this H&E stain. We stain the nuclei, which is what the H, the hematoxylin, stains the nuclei. The E refers to eosin. Eosin stains everything else, all the proteins. These fluorescent dyes are made to replicate the H and the E stain. Our cameras, however, are grayscale cameras. When we do fluorescence imaging, we use a black and white camera to collect the light. We have filters to filter out the different wavelengths, but the camera is monochrome. So to create these images, we false color them in software. And so we can do that using a simple Beer-Lambert law absorption physics-based model where we're modeling the absorption of light through these dyes, and we can color the tissues to look like their standard H&E, standard of care H&E. And so here's an example of a 3D data set of a prostate needle core biopsy. This is a 1 millimeter diameter plug of tissue. We're looking from one side to the other, slice by slice, from one end to the other. Here we have 12 biopsies laid out in a custom biopsy holder that we've machined, and we can image them one after another. You can see the detail. These are all nuclei, these round little purple donut-shaped things are nuclei. These are the glands. They form
Is
that
that
branching tree.
sorry,
Yeah.
John, is that moving in real time? I mean, we're seeing actual movement of the nuclei?
Well, this is a depth stack of images. So this is dead tissue. Let me clarify. This is
Okay.
fixed tissue. So nothing's living in this tissue. It's completely stationary. But the reason why it looks like it's moving is because we're slicing through it from one end to the other. I'm just showing a depth stack of images.
I get it. Thank you.
Yeah, it's like the branches of a tree that you're seeing kind of one cross section after another as we go through the depth of the tissue. So you can see that these glands are interconnected. They form this branching tree structure. But if we pause at any one moment, this is what typically they would see as a single cross section on a glass slide. And it can be misleading. For example, there's a type of Gleason pattern 4. You remember, pattern 4 means you have to get surgery or radiation typically. But there is one type of pattern 4 gland. They call it a poorly formed gland. And let me see if I can find an example. For example, here you'll see what looks like a gland that's sort of pinched off. It's not really hollow anymore. It looks like it might be a poorly formed gland, but that's just an artifact of where you cut the tissue. If you're looking at a 2D section, if you kind of look around it, you see, well, this is a fully formed gland. Of course, if you find the right depth, it looks poorly formed, but that's the type of artifact and ambiguity that you get when you're looking at a single 2D section. Of course, in 3D, now we have the full picture. And that could save the patient from getting surgery when they don't need surgery. So it can make a big difference for the patient. Here's another example of, this is actually mouse tissue, mouse kidney tissue. So you can see, we can label different structures, the gray nuclei. These are the tubules of the kidney. The kidney is a filtering system. This is the glomerulus, the primary filtering structure within the kidney, where the blood vessels kind of remove their toxins into the urine. And so here's actually a volume rendering. So instead of that slice by slice view, you can reconstruct it to look like a volumetric structure. Here's that glomerulus filtering unit. Here's a blood vessel poking out of one face, and then we can fly through it if you want to. Keep thinking it would be nice to get VR goggles for this and try to render this so people can fly through it themselves. And here's another example. As I mentioned, we do some imaging of mouse organs. This is a whole mouse brain. In this case, we used our latest system, and we started with the low resolution mode because within a few hours, we can image the whole mouse brain at low resolution. If we tried to image this whole mouse brain at high resolution, it would have taken us several weeks. It's just extremely slow. But here at low res, now we can see where the brain, these are metastatic deposits. So brain cancer that's metastasized into these mouse brains at different sites. And once we know where those mets are, we can define some regions of interest and go back and image those at high resolution and provide what they need to be able to quantify these structures. These are the high resolution views where you can see the nuclei, you can see every single cell, and start to quantify these tumors. So that's an example of a multi-resolution workflow. So in terms of challenges and opportunities, what we need to do to get this into the clinic is to prove that 3D pathology has benefits over 2D, which is the standard of care right now. And we got to do that in hundreds of patients, ideally, maybe thousands, to have that clinical evidence that people want. And computational analysis, of course, has to be part of the story because our data sets are so massive. We routinely generate on the order of terabytes of tissue, even from a single patient. 12 biopsy cores, for example, the raw data could be as much as a terabyte. And it's too much for a human to go through slice by slice. So we use AI, and there's two general ways to apply AI. One is sort of this black box CNN approach or deep learning approach, where you have the imaging data on one side and you have the patient outcomes on the other side. And you just use the black box to figure out how to associate the images with the patient outcomes. It can work very well if you have a lot of data that's well curated, that's well annotated, but it can be tricky, and you need to validate this very carefully. It's essentially a black box. You don't really know why it works. And so that's one of the problems for clinical adoption, at least by pathologists. They want physical insights. So the other approach is what we call a handcrafted approach. And what that entails is first segmenting out the structures that they already trust. For example, the glands in the prostate obviously are what they use to grade the severity or the aggressiveness of the prostate. So if we can segment out and extract out the glands themselves and quantify those glands and train a more traditional machine classifier, that will provide insights and be more palatable to the pathologists. And so we're actually doing both. But the first step for the handcrafted, at least, is to segment out the glands. And there's different ways to do that. Again, you can use deep learning to be able to segment, to create a segmentation mask. Basically, we want a binary image of where the glands are versus where they aren't. And we have our H&E analog data sets on the left. To be able to get from here to the segmentation mask, one way is to ask pathologists to manually annotate, to circle all these glands, and then train the computer to do that automatically. This is tedious and subjective, so it's not ideal. Another approach is if we can label the glands directly with an antibody. So these are specifically labeling just the glands. We can do that, but it's very time consuming and expensive. These antibodies are expensive and they're large molecules that don't diffuse quickly into tissues. Unlike our H&E analog, these are small molecules, dyes that diffuse into the tissues within minutes, within a few hours, we can stain the tissue. With large antibodies, unfortunately, it takes weeks for us to stain thick tissues. But if we could do that, then we can apply traditional thresholding, we call this computer vision based techniques to segment out the glands based on this very specific, molecularly specific label. So what we did ultimately was to train essentially a deepfake algorithm is the way to think about it. We have our H&E analog images. We can train a deep learning model to convert this image to look like an immunofluorescence image, but this is a synthetic immunofluorescence image. It is a deep fake, but it's very accurate because we train it in a fully supervised way with tissues that we've stained with our H&E analog plus the antibody of interest. We call this tri-label tissues, triple label tissues. And now we train the computer on how to convert from the H&E to the antibody stain. Once it does that, then we can use our traditional computer vision thresholding techniques to create the segmentation mask. So this is what it looks like in the end. You have your H&E images. We trained a synthetic model to create these synthetic immunofluorescence images. Now we can segment out the glands. The yellow are the cells, the epithelial cells that line the glands. The red is the lumen, the hollow centers of the glands, and the gray is the surrounding tissue. And we can do that for normal glands, benign glands, as well as cancerous glands. And you can see here, there's clearly a difference in the appearance of benign glands versus cancer glands. I like to say that benign glands are like a mature tree. There's hierarchy. There's the large branch, the large trunk. There's smaller branches and even smaller branches. And they're very mature branching tree network. A cancerous gland network looks more like an immature bush. Lots of small branches crowded together, densely packed, not as much hierarchy and diversity compared to a mature normal gland network. So that's how I usually describe the glands. This is what a prostate biopsy looks like in 3D once we've segmented the gland structure. So this, again, about a 1 millimeter diameter. We fit it into this rectangular channel to image it, so it's no longer has a cylindrical shape. But from here, you can see it's a very complex structure of glands, snaking through even this 1 millimeter core. You can see a lot of information. We can even extract the skeleton network. This is the center line that meanders through the gland network. And from there, we can pull out a lot of quantitative parameters to start to describe those glands. And so we did that with a preliminary clinical validation study. So here, this is a fairly small pilot study with only 50 cases. These are patients that were treated at UW 15 to 20 years ago. So about half of these patients had recurrence of their cancer after surgery within 5 years. We call that 5-year biochemical recurrence. Half of these patients, 25 of the patients, did not have recurrence within 5 years. So that's our high risk and low risk group. We're trying to see if we can predict which patients have aggressive cancer that returns within 5 years of their surgery, and which patients had indolent cancer that did not recur. So we pulled out simulated biopsies from these archived specimens that are in the tissue bank here. We stained it with our H&E analog, cleared it to make it transparent, imaged it with our OTLS microscope. We applied our gland segmentation algorithm to try to quantify the glands. And we compared 3D versus 2D. With 2D, they usually look at only 3 cross sections within the biopsy. And so we compared what would it look like if we try to quantify the glands by looking at only those 3 levels with those 3 cross sections within the biopsy versus looking at the full 3D network within the biopsy. And so we correlated these features with the patient outcomes. This is a pretty busy plot, but basically it's showing that with different parameters, for example, the curvature of the glands or the waviness of the gland walls, that's this gland to convex hull ratio, we can start to look at the performance of 3D versus 2D. With 3D here, BCR stands for the aggressive cases where they had biochemical recurrence of the cancer within 5 years. And the non-BCR were the patients that didn't have recurrence within 5 years. Here you can see in general, the curvature of the glands is higher for the patients that had recurrence. And that makes sense. As I mentioned, with cancer glands, you tend to have smaller glands. They're more densely packed. And so they're more curved. They're smaller, they're more curved. Also, here we see that the aggressive cases have less wavy walls in the glands. That also makes sense because the immature cancer glands tend to be smoother, whereas the mature normal glands tend to have these wavy walls, kind of like a mature tree with a very wavy trunk bark area. And here you can see that the 3D feature shows better separation between the aggressive and the non-aggressive cases. And that's shown here in this receiver operating characteristic. That's what the clinicians like to see. So on the y-axis, you have what we call sensitivity or true positive rate. And on the x-axis, this is the false positive rate. So in your ideal diagnostic, you want to have things as close as possible to this upper left corner. That means you have a high true positive rate and a low false positive rate. These two quantities trade off depending on how you threshold things. And so this curve shows us the trade-off between true positivity and false positivity. And in both cases here, you can see that the 3D feature gets closer to the upper left corner. It has this higher area under the curve. That's the metric that the clinicians like to see, is the area under the curve. We want to get that as close to 1 as possible. Another way to look at the data, well, before I get to that, here's if we look at multiple parameters. So we had about 12 parameters that we extracted from these glands. These are physical parameters like curvature, density, tortuosity, like how curved are they, how tortuous and twisted are they. So we took about a dozen of these features, ran it into a multi-parameter machine classifier. And again, you see with the 3D multi-parameter model, the area under the curve is higher by quite a bit compared to the 2D model. And then for some of these cases, we actually had time to recurrence. And so this is another type of analysis that the clinicians like to see. It's called a Kaplan-Meier analysis. The x-axis is time. So here we have about 5, 6, 7 years of time. The y-axis is what we call progression-free survival. So in this case, it's recurrence-free survival. At time 0, that's the surgery date. So this is the date that the patients had their prostatectomy. At that point, none of the patients have recurrence yet. They just got their tumor removed and they just got their prostates removed. And so there's 100% of them have no recurrence at that point. So they're up here. Over time, more and more patients start to have recurrence of their cancer. And so each time you have a drop in this curve, that means one of the patients had a recurrence. So if we run our best 2D model to try to stratify between high risk and low risk patients, you see that these curves are not very different from each other. Meaning it's hard for us to predict with the 2D model which patients will have recurrence versus not. But with the best 3D model, you can see these curves are very far apart. We can more accurately predict with the low risk group, very few of these patients have recurrence over time. But with a high risk group, you can clearly see a lot of them recur within 5 years. And so that's another way of showing that our model, our classifier, is good at being able to separate, to prognosticate between high risk and low risk prostate cancer patients. So in summary, this is a very multidisciplinary endeavor. Clearly, there's a lot of different technologies at play. There's chemistry. We have to clear the tissues, label them. We want to label them with antibodies. We want to look at nucleic acids in the future. We want to automate some of our tissue prep. The imaging, there's a lot of innovation there, designing the microscopes to be fast and accurate and high resolution, and applying them to different applications, whether it's preclinical or clinical, trying to integrate them with other genomics or other omics technologies like sequencing. But a lot of the opportunities and a lot of the work that we're doing now actually is on the data processing and analysis, just because we're generating this wonderful data that's hundreds of terabytes of data, but it's not going to be useful unless we can analyze it. And so a lot of the students now, they want to learn how to do the machine learning. So we're in a mechanical engineering department, but we've become much more computational over time. And so there's a lot of things we can do, including more segmentation of key structures. I showed prostate glands as one example. Well, people are also interested in segmenting the vessels or the nerves, because those are the highways within which the cancer cells tend to migrate out, and how they metastasize out of the tumors, either through the vessels or along the nerve tracks. So that's another thing we're trying to work out now, is to segment out all of those structures to create holistic classifiers for very precise decision support for the patient. And we're also doing the black box deep learning AI methods, where we just treat the data as without segmenting it. We use the raw data and then do deep learning to get straight to the classifier or the patient outcomes. And so there's a lot that we can do there as well. So let me just wrap up. I'm going to skip through this other example for the sake of time. I think this is repeating kind of what I said before, some of the work that we're doing on the computational side. Here's my final slide, kind of to sum things up. 3D pathology, first we need to create the data. So we have either external reference labs that the clinicians can turn to. They can ship their samples out. We're finding more and more pathology labs at various hospitals want their own microscope, and that's been good. The company's been selling some of these microscopes, and they just established a partnership with one of the biggest and most respected clinics in the academic clinics in the nation, Mayo Clinic in Rochester, Minnesota. So that's been a nice development. In terms of analysis, there's different risk levels. One would be pathologist interpretation of the data visually. This takes time, it's very slow, but in certain cases it may be fine if we have smaller tissues. That's pretty low risk because we're still relying on a pathologist. Medium risk would be to use AI to triage, to find the high risk regions or to find the high risk levels within the 3D data sets and to feed that to the pathologist. That should be very appealing to pathologists. They can stay in the loop. They're still responsible for interpreting the data, but the AI does all the hard work of getting rid of all the simple cases. There's a lot of obvious cases we can trust machine learning. The challenging, ambiguous cases we can send to the pathologist. And finally, full AI, as we talked about, if we can develop full computational models to start to guide treatment decisions, to identify the aggressive cases or to identify which patients would respond to which drugs, that would be what we want in the future. And 3D pathology alone is not enough. We want to combine that and integrate that with sequencing, molecular assays, radiomics, machine learning of radiology images is also important. And ultimately combining all of these different technologies to provide enhanced decision support, both for clinicians and patients. That's the vision for the future, to improve and modernize the field of pathology, which really hasn't changed too much over the last 100 years. It's still regarded as the gold standard, but we're trying to bring it into the digital era, bring it into the 3D world. So with that, let me just thank and acknowledge some people, especially Adam Glazer, as I mentioned. He was the postdoc who built a lot of our first systems. He's now at the Allen Institute working on neuroscience applications. Wei Xiao is a former PhD student in my lab who did a lot of the computational analysis of the prostate glands that I mentioned. And most of our funding has been from the NIH. Got some funding from the DoD, Department of Defense, some other foundations as well, and working with people all over the world at Oxford, at Emory, and it's been a fun ride. So thanks for the opportunity to present. Happy to take any questions.
Okay, do we have questions? I know I had a couple questions. Maybe I'll just start with mine. And if you do have questions, go ahead and maybe unmute and give that. But let me go ahead and start with one. How do you train going to this training of the AI? How do you do that? You said the black box approach. How is that done? Just kind of the wide open question. I guess I don't know much about that.
The short answer is I don't know, because I'm not the computer scientist who invented the deep learning models. There's been a lot of work that's been done over the last few decades to develop these neural networks, and they are just a series of convolutions. They're down sampling the data, connecting the different pixels. You know, when you have an image, it's basically an array of values that it represents the intensity of each point in the image. And so they're trying to figure out relationships between different pixels within the image that persist for diseased tissue versus non-diseased tissue. So the first step for any deep learning method is what we call labeling. You first need the labels. For example, in our case, it's these patients have aggressive cancer, these patients have non-aggressive cancer. So these images we feed into the deep learning model, we give it that label, we say, okay, this belongs in one camp. And these images are from the patients that didn't have aggressive cancers. Now the deep learning algorithm is starting to learn how to associate the images to the patient outcomes. And exactly how it does it is beyond me, but we have to, of course, validate it very carefully. So as long as we can do the training well, and then we take an independent validation set that the model has never seen before and show that it can predict accurately on that validation set. That's basically what a lot of people are doing now is training and then validating carefully that the models are working. But they keep improving these models. For example, 5 years ago, these convolutional neural networks were kind of like the state of the art for doing the deep learning. Now there's something called transformers, image transformers, vision transformers that are being used, which are actually used for natural language processing. So when you speak into Siri or Alexa, and it's listening to your words and interpreting it, they are often using these transformer networks. And so they're starting to apply those to the image analysis as well. But again, I'm not a computer scientist, so I can't give you the full story on that, but
okay,
we certainly use the tools.
Yeah, thank you. Other questions?
Yes. Can I have something?
Yes.
You started out by classifying your study with PSA levels, and at the end you went into ranking your 2D and 3D success rates against the high risk and low risk people. Is that the overall theme that went through your selection process, or is there some other way that you were ranking the high risk versus the low risk prostate cancer subjects?
Yeah, so for all of the clinical study I talked about, when we looked for the right patients to pull their tissue, archived tissue samples, we were selecting based on their recurrence outcomes. So there was a previous study that was done at UW where they recruited hundreds of patients, and about half of them had recurrence of their cancer within 5 years of their surgery, and half of them didn't. So we tried to get that same 50-50 mix so that we can train the classifier effectively. So it's all based on those what we call biochemical recurrence outcomes.
So you were ranking this against the 2D and the 3D classification you presented in the latter slides. Now, have you had the opportunity? Is that because you're using your technology and your techniques with those 2D and 3D, or is that some other earlier development of 2D and 3D assessment of prostate cancer?
Oh, I see. No, that's with our technology. So up until now, the technologies have not been there to be able to collect that 3D data. So this data, the data sets were created in our lab. They're very novel, and actually we post them online. They're publicly available now through the NIH, and we're hoping other people can use that data to also analyze the information that we've provided.
So is this technique that you have, or the methods that you're using with your equipment, is that now readily available, or is it only just the very few, such as the Mayo Clinic has the opportunity to use not only your equipment but your evaluation methods?
Yeah, so the technology, the hardware is now available. I wouldn't call it widely. The company was started a few years back, and so they probably placed a handful of devices now. They have orders for maybe a dozen more. So it's growing rapidly, we hope. But that's all over the world. So most clinics are not going to have it anytime soon, I would say. The other part, the computational analysis will take even longer because that will probably require some sort of regulatory oversight. And so we published our paper, but as I mentioned, that's a very preliminary study with only 50 patients. In order to get this through FDA, for example, would probably be, well, I can't put words into the FDA's mouth, but probably we're looking at closer to 1,000 patients to provide that level of rigor so that we can publish it in the best journals and get the clinicians to adopt. But there is a path also to get adoption of these types of assays without FDA oversight. For example, a lot of these genomics tests are not necessarily FDA approved. They go through this laboratory test approval process called CLIA. It's a laboratory developed test, they call it, an LDT. So there is a pathway potentially to being able to get some of these assays out sooner without the long process of FDA approval. But yeah, we'll have to work through that in the years to come.
So are you the manufacturer and sales of these devices here in Wallingford, or are they assembled by somebody else?
Yeah, right now they're all assembled, manufactured here in Wallingford. So yeah, it's the parts are purchased from various different places, but it's definitely assembled together, aligned. Yeah, there's some custom parts, there's some off-the-shelf parts. Cameras, for example, we order from different companies, lenses from the other companies, but it's all kind of put together in the company here.
So do you then have a sales and service organization that takes care of these, or are these one-off and people have to maintain the operation on their own?
No, yeah, with these types of microscopes, there's almost always a service contract that's required, and actually most people who buy these want that because the alignment, the maintenance, yeah, you definitely need very skilled, trained technicians to be able to support that. So yeah, they put that into the purchase agreements is the ongoing, and that's an ongoing source of revenue for the company, of course, as well.
Sounds like that's the secret sauce right there.
How much do these cost if you want to buy just one?
The microscopes, I believe they're selling them for about $500,000. Yeah. But they're not cheap. But compared to similar microscopes built by the big companies, it's actually, that's on the low end, I would say. Some of these other 3D microscope, confocal microscopes, tooth photon microscopes can be as high as $1 million or more.
So are you in competition with a different 3D microscope then? Or is this a unique product that you have?
It's fairly unique. There's a certain niche. Yeah, there are microscopes that have much higher resolution. They're designed for biologists that want to see very fine structures, but they're going to be slow for clinical specimens, and the sample mounting is not ideal. So I feel like for clinical specimens, we don't have a strong competitor right now, although other people are probably trying to do similar things. But I think we have the early mover advantage there. We've become known. Our lab basically was one of the first to apply the technology to clinical applications, and then the company, of course, formed around that. And so I think that is an advantage right now.
Golly, I think with that, let's go ahead. Is there any quick questions? We're already running over, so I want to thank John so much for this presentation. Well, let's go ahead and cut it right there, and if we can, just hand clap, although everybody's on mute. So thank you, John, so much for
Oh yeah, no problem.
talking about
and if
this.
there's ever any questions, feel free to email.
Yeah.
You can look me up.
Is there a website for your company? I mean, that might get some of John Jacobson's questions.
Yeah, if you're interested in talking to the company, Nick Reeder, I could put you in touch. They have a website, although it's not super detailed. They're early stage, so it's alpenglowbiosciences.com. It's pretty long website.
Alpenglow.
Alpenglow Biosciences. Let me pull up the name. So here, ALPENGLOW Biosciences, all one word, .com.
Alpenglow Biosciences.com. Okay. Well, I'll have to look at that, and I guess
yeah, feel free to send that out.
Yeah, and John, it sounds like you may have follow-up questions, but maybe take a look at that, and that'll probably answer some of them, and then follow up.
I'm just amazed.
It is amazing.
I remember the suggestion halfway through my PhD study. I was suggested that I look at using light technology rather than ultrasound technology, and I didn't quite have the guts to quit halfway through my project and start over again with something I didn't understand. So I'm just totally amazed and very appreciative of your taking the time to show that to us.
Yeah, you're welcome.
Thank you very much.
And if anyone ever wants to see the devices in action, let me know. I'll give you a tour of the lab.
Oh, wow. That would be fun. We might do that. I'm gonna go ahead and stop the recording here. So stop the recording.